I'm starting recording right now.
Okay.
Okay, so welcome everyone then. I will just give a very short presentation of Maria Blasco. She is one of the world leading figure in the study of telomeres, and she has focused in particular on the complex interactions of telomerase and telomere length, in particular in the context of aging, of cancer, and of other age-related diseases. So she has more specifically developed and also masterfully exploited a family of mouse models characterized by short telomeres, and also a series of techniques related to the observation of telomeres, telomerase activity, and so on. And in the last 10 years, but also before, of course, she has published major research articles in the greatest journals. I'd like to say that she has started a career, she'll tell us maybe what she thinks about that, but that's my opinion, at a moment where when some were still advocating a very simplistic view on the role of telomere shortening in aging. And she has actually been a major contributor to the now much more qualified and articulated picture we have on this so-called hallmark of aging. A recent result, for instance, that illustrates this is the discovery in a lab that if telomere length is not associated to longevity across species, it is, on the other hand, likely to be associated with the rate of telomere shortening. So that's one of the results of a lab among many. I also want to highlight the clarity and the sobriety of the reviews she wrote as a first or as a last author. Something I appreciate a lot, and I think is to be appreciated, is that there is no unjustified promise of a magic treatment or a complete explanation of the phenomena that are investigated. And that I think is very precious. She's currently head of the Spanish National Cancer Research Center, the CNIO, and we are of course very grateful that she accepted to pay us a virtual visit, and of course we are also frustrated that this visit is only virtual, and we hope to be able to do better in the future. So thank you again, Maria, and the floor is yours.
Thank you very much for the very kind introduction. I'm very happy to be here with you using teleconference. I think this is what we can do now, and it's not working so bad. I think it has been quite useful during this crisis. So I will give a very general talk, and we can, in the questions, we can go into more specific questions, because I see that you are quite familiar with the work that I have been doing. So I think my main interest as a scientist is really to try to eliminate disease, or to be able to fight disease. And we know how to do it. This is the work of the end of the 19th century, early 20th century, with infectious diseases. We know that the only way to cure a disease is really to understand its origin. And we have done this with the smallpox, with bacterial infections, with the antibiotics. HIV is a great example of how using molecular biology, we were able to really be able to control a disease like the HIV. Then we had Ebola, now we have the coronavirus, the COVID-19 disease, and we know that the way to go is really to understand the origin. In the case of other diseases, which are not infectious diseases, like the diseases that are killing us today, the majority of deaths are produced by diseases that are not infectious diseases, but are diseases associated with aging. So I put here several of these, like cancer, cardiovascular, diabetes, different degenerative diseases. We know that these diseases have a common cause. This is not a virus or a bacteria in general, but is the process of aging. And I will put some examples, for instance, with degenerative diseases like fibrosis, but I will also talk about cancer extensively at the end of my talk. But basically the idea of my lab is that in order to be able to cure these diseases someday, or to prevent the diseases, we have to understand the cause, and the cause is aging. So we have to understand aging. So this is some examples that really aging is the cause of these diseases. This is the incidence of cancer in men and women. These are data from the Cancer Research UK. This is in the UK, but the same is in Spain or France or in any developed country. You can see that cancer is quite a rare disease when we are young, but as we age, the incidence of cancer increases. We can even say that it's very rare to have a cancer when you are less than 40 years of age, and this increases very significantly after 40, 50 years of age. The same can be said for heart failure. These are again men and women, and you can see that to have a heart infarct is quite rare when you are below 40. But after 40 years of age, men and women, they can develop one of these heart infarcts. So this is telling us that there is something, some biological mechanisms that are underlying these diseases, which are of course related to aging. This is not only a biological problem, a scientific problem, it's also one of the challenges of humanity, because this there is a phenomenon which is different from the biological aging, that is called demographic aging, that is increasing the number of people that are at advanced ages in the world. And this will mean that the diseases associated with aging will increase dramatically the incidence. These are the projections for Alzheimer, but the same can be said for cancer or other diseases associated with aging. So it's really important to understand what is aging. So this is our view of what is aging. We think that when we are young, our cells, let's say, are not aged, and this is preventing us from developing different diseases. But when this molecular aging occurs, this aging is really the cause of the diseases. The different diseases are the consequence of this aging phenomenon. So they have a common cause, which is this aging. If we know now that aging can be produced by the genes in part, so there is a genetic component, but it's also very important the habits, the environment, the interaction with the environment can also determine this rate of aging. In any case, if we know these molecular causes of aging, we will be able, on one hand, to have biomarkers, for instance, that would allow to tell, how different people are aging at the molecular level, and this will be a way to understand the risk of a given individual of developing different diseases. But in theory, we know which are the causes of aging. We will be able to intervene and design molecular interventions, therapeutic interventions that should be able to delay aging, to increase the time of life in which we are healthy, we are young, our cells are young, and delay different diseases. And this is the approach that scientists are taking in order to find which are these molecular causes of aging. One of the tests that we do in the lab is to see whether we can extend the lifespan of a model organism, whether we can extend the lifespan of a mouse, for instance, whether we can make a mouse to live younger for longer times. And this is telling us which are these molecular pathways of aging. Even though the final goal is not really to live young forever, but to be able to find cures for these different diseases, we usually analyze the impact of different molecular pathways in aging by studying longevity. So this is a sort of a change of paradigm, because what this is telling us is that disease doesn't appear when you go to the doctor and the doctor tells you you are sick. Disease may be going on in your organism, in your body for decades. And this is very clear now. For instance, if you look, for instance, at cardiovascular disease, and in particular the presence of plaque, for instance, in the different arteries of the body, which would lead to a heart infarct, maybe in the future. This is a study that was published in 2015 in the institute near the CNIO in Madrid. You can see that already at 40, 50 years of age, both men and women have already these arteries significantly blocked. So even though they are healthy, they don't have an infarct yet, they are completely healthy, men and women, there is already disease, which we are not seeing. But if you look with detail, you can see that this disease is happening in our body. And I don't know if to call it disease or not, but certainly abnormalities or pathological abnormalities are occurring in our organism way before the disease is diagnosed. And this is very important. Of course, scientists think, well, this aging phenomenon is something very robust. It must be very difficult to change the way a human being is aging. But if we look at nature, and this is a review which is quite interesting, probably you know it already, and you look at the different longevities of different organisms, you see that longevity and aging is very flexible in nature. And you can have organisms that live very short times, for instance, a mouse, or you can have plants that live up to 8,000 years, 80,000 years, or even some organisms that could be considered immortal because nobody has been able to see them age, like the hydra, for instance. So it's interesting, for instance, to see that species which are very close genetically, like these mammalian species that you see there, like the mouse, the neck mole rat, and the little brown bat, they are similar in size, they are all mammals, but they can have very different longevities. And this is telling us that it must be very flexible, this aging process, and we can manipulate it. Here we are the humans. We are not the longest lived mammal at all. The whales can live longer than us. And then, of course, you have the turtles and oysters, and even this shark, the Greenland shark, which can live up to 400 years. I mean, this is something amazing to think, right? So these individuals are living in the seas for 400 years, and basically they are young during this time, right? This is just an example, more or less. There was found a shark that it had 420 years, more or less, the same time as where this expedition from Magallanes and Elcano, made the first global circumnavigation. So, longevity can be very, very extended. So, which are these molecular causes of aging? This is a review that has been quite influential. That I contributed to this review, and it's called The Hallmarks of Aging, in which we describe which molecular pathways have been shown to have an impact in longevity. Because when you manipulate these pathways, for instance, in mouse models, you can either shorten the life of mice or extend the life of mice. And one of these molecular causes of aging is the shortening of telomeres. So telomeres are structures that are at the end of the chromosomes. They are very important to protect our chromosomes from rearrangements. So if the telomeres are destroyed or they are too short, the telomeres become unstable and they can fuse to each other and generate chromosomal aberrations. And this has been shown to be one of the molecular pathways of aging. We think that is a primary cause of aging because when telomeres are short, this triggers other molecular causes of aging. This triggers genomic instability. This impairs the ability of stem cells to regenerate. It leads to cellular senescence, to mitochondrial dysfunction, to very vast epigenetic alterations. So a single event like the presence of a short telomere can trigger different molecular pathways of aging. So we think this is one of the causes, one of these germs, if you want, of the diseases associated with aging is the shortening of telomeres. There is an antidote, there is an enzyme that is able to compensate for this telomere shortening. It's called telomerase. Telomerase usually is present in the early embryonic development, in the early stages of embryonic development, such as, for instance, the blastocyst stage. And there, telomerase is activated. It can elongate the telomeres of the new individual, of the embryo. And basically, it's thought that with these telomeres that are elongated, this individual will have to live his or her life, right? Depending on the rate at which this telomere shortening happens, diseases may appear earlier or later in this individual. So this is the theory, right? So we have, this is the idea. So the telomere length would be decreasing with aging. This will happen in all organs and tissues, and this also includes the stem. Sure, this would trigger, do you hear me? This would trigger.
Yes, you were interrupted for a few seconds, but now we can hear you.
So the idea is that short telomeres would trigger disease, would be at the origin of disease. We have now quite a lot of evidence that this is the case. I did when I was a postdoc with Carol Grader. And I will tell you also about a number of diseases that were discovered after that, after the work we did with mice, and I'm sure that this could be true. There were a number of diseases found in humans that are called telomere syndromes. These are individuals that have mutations in telomerase gene, or genes that are important to maintain the telomeres, and they are going to die prematurely because of premature loss of the ability of tissues to regenerate. And they can develop diseases like dyskeratosis congenita, aplastic anemia, or different types of fibrosis, like pulmonary fibrosis, renal fibrosis, liver fibrosis. Fibrosis is an end stage where the cells, basically the cells cannot regenerate, and the tissue cannot have a normal structure. And instead of having the normal cells in the tissue, you have fibroblasts that produce scarring, and this is called fibrosis. And this is an end stage of the, different tissues, like the lung, the renal, the liver. So this was telling us that short telomeres were at the origin of disease. We know now that even people that are normal and they don't have mutations in telomerase, for some reason or other, they can have a faster rate of telomere shortening. They can end up with shorter telomeres than normal. And this has been shown to be correlated with a higher risk of disease. So even in the absence of telomerase mutation, presence of short telomeres could be indicating a higher risk of disease for different individuals. So as I said, the first time that this was demonstrated actually was with the generation of telomerase deficient mice. So we generated mice that lack telomerase. So this is an artificial system in which we force the telomeres to get really very short because we remove the telomerase gene. So these mice have much shorter telomeres than normal, and it was the first time that it was actually demonstrated that telomeres were important for aging, that telomerase was the enzyme that was maintaining the telomeres in mammals, because this was not formally proven. And basically all this work demonstrates that short telomeres are sufficient to induce aging. So if you really shorten the telomeres, this is going to induce diseases associated with aging. And this is happening because short telomeres impair the ability of stem cells to regenerate our tissues. So if the telomeres are short, our tissues, which accumulate damage all the time, will not be able to cope with damage, will not be able to recover a normal tissue homeostasis. So this was mentioned in the introduction. So we wonder, well, this has been shown in humans. We have the telomere syndromes in mice. We have shown that the telomeres are short. This impairs longevity. But this is a general phenomenon in nature. Are telomeres really something that evolution has worked on to determine the longevity of different species? And last year, we actually collaborated with the Zoo Aquarium of Madrid and also with a group in Barcelona working in seagulls in the Delta River, in the Ebro Delta River in Catalonia, in Spain. And we decided to measure the rate of telomere shortening in different species of birds and mammals. We didn't include everything. So this is limited only to birds and mammals, but birds and mammals are quite distant in evolution. So they are quite distant. So we measured the rate of telomere shortening. We saw first that telomere length at birth was not correlated with longevity. So, and we already knew that because, for instance, mice, when they are born, they have much longer telomeres than humans. But a mouse lives much shorter than a human. So a lot of people were saying, well, this means that telomeres have nothing to do with aging. Well, it's not true. When we measure the rate of telomere shortening in humans and mice, we see that even though mice are born with much longer telomeres than humans, they shorten the telomeres 1,000 times faster than humans. So we already knew that maybe it was not the length, the telomere length of a species what determined longevity, but could be the rate at which this species is losing telomeres. And I'm not going to show the whole paper, if not we wouldn't have time, but this is the conclusion of the paper. So we found that different species have different rates of telomere shortening. This could be adjusted to a power law in which basically just by knowing the rate of telomere shortening, we could infer the longevity of a species. Something interesting here is that you could choose two species, like for instance, an elephant here and a flamingo. Elephants and flamingo are very distant in evolution. So an elephant is a mammal, is closer to a mouse than to a flamingo. However, the elephant and the mice, they have very different rates of telomere shortening, even though they are mammals. And they also have very different longevities, right? A mouse only lives 2 years, an elephant can live up to 80 years. So the genes don't seem to be so important here to determine the longevity of a mammal and an elephant, of a mouse and an elephant. However, the rate of telomere shortening is important, because if you now compare an elephant and a flamingo, even though they are evolutionarily very distant, they have the same rate of telomere shortening. And they also have the same longevity. More or less, a flamingo and an elephant, they have a similar longevity. So this was very interesting for us, because it seemed that something that is more of an epigenetic trait, like could be telomere length, which could be influenced by the genes, but it also can be influenced by the environment, was important to determine longevity. It was not so much the genes, it was the rate of shortening of these telomeres. So all this is telling us that telomere shortening is important for aging, and probably has been something that evolution has been using to determine the longevity of different species. But we wanted to go one step further, and we wanted really to see whether we could delay aging, we could challenge the longevity of a species by manipulating telomere length. And the first time that we did this was, we did this genetically. We generated a transgenic mouse, so a mouse that had telomerase overexpressed throughout its life. And at the same time that we put telomerase, we also increased the copy number of genes that are tumor suppressor genes. These are genes that eliminate damaged cells like P53, P16, P19R. So we made a mouse that was cancer protected and also had more telomerase. And when these mice, we call them triple because they have different genetic manipulations, we were able to see that we could increase a lot the median longevity of the species. So in these mice, two things happened. So we found that the health span was increased. So the first mouse that died much later than the first mouse that dies in the control group. In mice, more or less, after 1 year of age, the mice start dying of cancer and different aging diseases. So what happens in humans in 40 years, in a mouse happens in 1 year. And these mice start dying. But if we manipulated telomere length, we could increase the health span, and the first mouse was dying much later. And on average, they had an increased median lifespan. Here, the maximum lifespan was not increased. And we think this is because of the small n we have here. We only have 12 mouse. And the maximum lifespan is provided by the outlier. So the maximum lifespan is provided by the mouse, which is weird, right? The one that has an increased longevity. And here we don't have enough mice to see this maximum longevity to be increased, because I will show you that we have done other experiments. You will see later, where manipulating telomere length, we increase not only the median lifespan, but also the maximum lifespan. So you can see here these mice, they are both old, 2 years of age, but the one that has long telomeres is younger in everything. So you can look at the paper, and we characterize many different things in the mice, and they are basically younger with the same chronological age. And that's why they've lived longer. Of course, you cannot do human transgenic mice, but you can try to find therapeutic strategies to activate telomerase in humans, for instance, or in mice, and see whether this can have the same effect. I mean, could it be possible to activate telomerase in an adult organism? And would this be sufficient to increase the lifespan, to delay disease? Would this have any deleterious effect, like increasing cancer, for instance? And we did this experiment in 2012. We decided to use gene therapy vectors. So, there are these vectors called adeno-associated vectors, where you can introduce a gene. These vectors are non-integrative, so they are not going to integrate and mutate the genome. They are going to be diluted out as the cells divide, but they will allow for the expression of whatever gene you put inside for a few cellular division cycles, right? So we thought this was perfect because we could put telomerase in an adult mouse. This will allow telomerase to be expressed. This will elongate the telomeres for a while, for a few cell divisions, and then the expression of telomerase will be lost because these vectors are non-integrative. They don't integrate in the genome. And in the case of telomere shortening, you don't need telomerase to be active all the time. You just need telomerase to be active a few cell divisions in order to recover telomere length, and then this telomere will be sufficient to allow for the tissues to regenerate, and you will be able to really delay disease. This was our idea, and we did experiment. We took mice, we gave them a single injection with these vectors, intravenous injection. This allows telomerase to be expressed, and then we saw that this was sufficient to delay many different diseases. So they had an improved glucose tolerance, less cognitive decline, less osteoporosis, cancer, which was something that we were worried about. We were worried that maybe by putting telomerase, cancer would increase, but cancer was also delayed. So the mice actually had cancer, but cancer appeared later. So was there any negative effect of having telomerase? We didn't see any negative effect. Actually, the mice lived longer. So both in the 1-year-old group and the 2-year-old groups, you can see here, we saw an increased survival, both median survival and maximal survival. So this is telling you, if the mice are living longer, this is telling you that having more telomerase by this gene therapy was not deleterious, right? Because it was increasing the survival of the mice. Anyway, we were very, very worried about this thing of elongating telomeres and to see whether this could have any bad effects. And we came up with a different way to elongate telomeres that we discovered by chance in this case. In my group, we were very interested also in understanding what happens when you go from a differentiated state to a pluripotent state. I imagine you are familiar with this induction of pluripotency that was discovered by Shinya Yamanaka and received the Nobel Prize of Medicine or Physiology. Basically, you can go from a differentiated state to a pluripotent state. It's like reverting what happens in development. In development, we go from a pluripotent state in the blastocyst, in the inner cell mass cells, to a differentiated state. And here we see telomere shortening as the cells differentiate and we age. However, when you induce these iPS cells, induced pluripotent stem cells, they are like these cells that we can derive from the inner cell mass. They are pluripotent. And we knew that when you generate these pluripotent cells, there is a high telomerase expression, the chromatin of the telomeres is open, so telomeres can get elongated. And when we discovered this, we saw that the telomeres of these pluripotent cells, they didn't stop growing, basically. So as you passage the cells, the telomeres kept growing. And we could end up with cells which were pluripotent that had much longer telomeres than those that you can find in nature, in the blastocyst. So we call these cells, these telomeres, ultra-long telomeres. And then we thought it could be interesting to use now these cells to try to generate mice that are born with much longer telomeres than normal, right? So instead of putting telomerase to elongate telomeres, we are going to manipulate the embryo and use these pluripotent cells with long telomeres that we have generated in vitro, basically just passaging the cells. We took A-cell morula embryos, we aggregated cells which have either normal or ultra-long telomeres. We can follow them with a fluorescent protein like EGFP, and then we can generate embryos that are derived from these cells with long telomeres. These are chimeric embryos. And from here, we can generate chimeric mice that are going to have some of the cells with a normal length of the species. This, for instance, a section of the eye. You can see here this area, which is not green. This is the normal telomere length in the cornea, in the eye. But you can see this area in green. The telomeres are much more intense. You see the telomeres are the red dots. These are areas of the cornea that are coming from these cells with ultra-long telomeres. So we have chimeric mice full of patches of cells which have long telomeres and others that have normal telomeres. And when we did this, we saw that we wanted to see whether this was good or bad. And we saw that it was not bad, certainly, because the mice live longer and they were cancer protected. So again, if you have an organism with much longer telomeres than normal, in this case, by using this trick that we use here, is an epigenetic trick with no genetic manipulations, we could, this was good for the mouse. So these mice had long telomeres and this protected against cancer. This is telling us again that cancer is the result of aging, I mean, of having more aberrations in our DNA, which are contributed by different types of damage. And one of these damages is short telomeres, because if we maintain telomeres longer, there is less cancer. We managed more recently, and we published this last year, to generate mice that were 100% derived from long telomeres. So basically, every cell in the mouse had very long telomeres. And we wanted to see whether this was good or bad. And again, it was not bad at all. We saw that these mice had actually less obesity with aging. They had a better metabolic aging, so they had a better glucose metabolism. They were also cancer protected. The mitochondria were working better. Everything was better in these mice. And these are mice that every cell in their organism has much longer telomeres than normal for the species. So I think with this, we are demonstrating that if having longer telomeres than normal in a species is not something bad, it's something that can delay disease and can increase longevity. So in other words, so short telomeres to disease, because if we are able to really intervene and make telomeres longer, we are able to delay disease. So with this, we decided to go and see whether we can actually have an impact on different diseases. So, I mean, we don't want actually to make humans to live longer per se, but to be able, remember, to cure diseases associated with aging. And here our idea was maybe we can use telomere gene therapy to treat diseases associated with aging. Maybe if aging is the origin of these diseases, if we are able to revert aging or to delay aging, we can stop the progression of these diseases. And we use both age-related diseases and these telomere syndromes, which are these extreme short telomeres that cause aplastic anemia, pulmonary fibrosis, etc. And sorry, I will talk about one of these diseases. It's called pulmonary fibrosis. Pulmonary fibrosis is the frequency increases with aging, the risk increases with aging. Is more frequent in men than women, and there are risk factors like smoking, pollution, etc. The interesting thing here is that there are families with a higher risk of pulmonary fibrosis, and it was found that the gene responsible for this increased susceptibility was telomerase. So if you have a mutation in telomerase, and you have shorter telomeres than normal, you have a higher risk of pulmonary fibrosis. So which was pointing to telomeres at the origin of this disease. But we know now that even in these are the papers that describe this, but even in the sporadic cases, which are not associated with mutations in telomerase, individuals with short telomeres, individuals with pulmonary fibrosis have short telomeres than normal. So this was telling us that somehow short telomeres must be this germ, this origin of pulmonary fibrosis. It may interact with other damages like radiation, pollution, smoking, in producing damage in the cells in the lung that are supposed to regenerate the lung, like the alveolar epithelial cells. But if you have short telomeres in these cells, we knew, our group and others have shown, that the cells with short telomeres cannot regenerate. So if you accumulate damage in these cells, but these cells have short telomeres, this is going to impair the ability of the lung to be regenerated. And this is something that is going to happen as you age, that the telomeres are going to become shorter. And if you don't regenerate, then you are going to accumulate senescent cells, DNA damage, and this is going to induce this immune response, fibroblast recruitment, and you end up with a scarring of the lung, fibrosis. And this is a lethal disease. So all individuals with pulmonary fibrosis, they die. There is no treatments that work. Actually, the treatments that are being used today in the clinic, they don't work. They can improve a little bit the life of the patient, but all the patients are going to die. So they are not curing. And we think they are not curing because they are not fixing the problem, which is the short telomeres. And you cannot regenerate. So we actually have shown this by doing the experiment. So we generated a mouse with pulmonary fibrosis associated to short telomeres. And then we treated these mice with telomerase and see whether we could cure the mice. We could stop the progression of pulmonary fibrosis and cure the mice. So actually, in this case, the mice are the patients. We induced fibrosis. I'm not going to go into the details. You can check the paper. We induce fibrosis. We detect which of the mice have fibrosis by CT, with imaging of the lung. And those mice that have fibrosis or an abnormal radiographic image of the lung, we treat them either with the telomerase or with a placebo, with the empty viral vector. And we saw that you can look at here, right? The mice that were treated with the placebo, 100% of them develop severe fibrosis. You can see the lungs full of fibrosis. This should be a normal lung, for instance, and this is an abnormal lung. But the mice that we treated with telomerase, so they had fibrosis and we treated them with telomerase, you can see that 50% are cured, no fibrosis, and 50% only have small patches. So this is telling us that this is an intervention that can actually stop the progression of fibrosis or can cure fibrosis. This is something that has never happened so far because my hypothesis is that we have not been able to cure fibrosis because we are intervening too late. We're intervening trying to prevent fibrosis, but we are not intervening at the origin, at the presence of short telomeres. And only when we intervene here at the short telomeres by increasing telomerase, we can actually rescue everything downstream, and this stops the progression of fibrosis. So this was the example I wanted to put you. We have done this with other diseases, aplastic anemia, heart infarct, etc. So one more thing, we were worried again about cancer. So are we going to cure maybe a fibrosis in a patient by putting telomerase, but then this patient, 10 years down the road, will develop a cancer in the lung? And in order to address this, we did an experiment in mice in which at the same time that we put telomerase, we activate an oncogene. So we are going to take mice, deliver telomerase into the lung, but also increase an oncogene and see whether telomerase could be helping the oncogene to induce more cancer. So this was our, if you want, our safety experiment. And the results were very clear. Putting telomerase was not increasing cancer, was not increasing the number of tumors, was not accelerating the time at which tumors appear. Basically, cancer appeared produced by the oncogene. This is something that we already knew. And telomerase was not having any impact. So it was not helping the cancer to grow because cancers already activate telomerase when they are induced with oncogenes. So we are doing now, we were generating the human vectors in order to be able to treat humans and show whether we can cure a human patient with fibrosis with telomerase. If that is the case, it would be actually the first type of therapies that are based on understanding aging in order to cure age-related diseases. So just the 5 more minutes, if I may, will tell you about cancer. So I've told you, remember, that cancer is an age-related disease. So remember that I told you that if we delay aging, we delay cancer. And we have shown this by manipulating telomere length. We have manipulated telomere length with telomerase, with a gene therapy strategy, but we have also manipulated telomere length epigenetically, manipulating the embryonic cells. And in both cases, when we generated mice with longer telomeres, they had less cancer. So cancer in part is produced by aging. But now imagine that, you know, once cancer appears and you have a cancer, you want to have a treatment and be able to cure the patient. And in this case, it's important, and I'm sure you're very aware of it, that one of the hallmarks of cancer is the ability of cancer cells to divide immortally. So this is a property of cancer cells. Our normal healthy cells are mortal. They divide a few cell divisions and then they enter senescence and they cannot divide anymore, or they die by apoptosis. But cancer cells, they are able to activate telomerase and they are able to divide indefinitely. So here my lab is also working in trying to kill cancer cells by destroying telomeres. So one possibility would be to inhibit telomerase, and this has been done, and the results have not been very encouraging because probably when you inhibit telomerase, you still need a time until telomeres shorten before you see really a shrinking of the tumor. And this may be too long. And actually the clinical trials that have been done with telomerase inhibitors have failed and have to be interrupted. So we forgot about this, and our strategy in the CNIO is not to inhibit telomerase, but directly to destroy the telomere. And we can do this because the telomeres, the structure of the telomere is the telomere is protected by a number of proteins. They are called shelterings. My lab has done knockout models for all these shelterings, and we have shown that if you remove some of these shelterings, you induce telomere damage in all chromosomes. So you can induce a massive telomere damage, and this is sufficient to stop the progression of tumors. We have shown this in a lung cancer model, and we have shown this more recently in a brain tumor model. So we induce brain tumors in the mice, and once the brain tumors are in the mice, we remove one of these proteins of the telomere. So we destroy the telomere protection function, and this stops the progression of the tumor. So in the CNIO, we are now developing drugs that are able to, we have screenings to try to look for drugs that are able to induce damage in the telomeres, and in this way to be able to stop the progression of cancer. And in this process, we have found something very interesting. We have found that many important cancer pathways actually are regulating this protein. So cancer cells really need to make sure that their telomeres are protected in order to be able to be cancer cells. And we have found very important cancer pathways like the PI3K-AKT pathway. First, we found this first, but now we have found many different cancer pathways like the RAS pathway, the AKT, the mTOR, many pathways that are actually making sure that cancer cells have the telomeres protected. So this is encouraging us because, and you can see here one of these telomere binding proteins with all the post-transcriptional modifications by these important cancer genes. So this is telling us that that cancer cells have to make sure that they have the telomeres protected. And this is an opportunity because we can now develop inhibitors against some of these enzymes in order to be able to prevent that cancer cells have telomere protection, and maybe come up with new types of cancer treatments which are based on targeting the telomeres per se, not telomeres, but targeting the telomeres. And we are working on that on the CNIO. We have a drug discovery program, and we are trying to find inhibitors that specifically disrupt the telomeres and to combine them with other inhibitors to be able to see whether this can be effective in the treatment of cancer cells. So this is I will finish here. This is my research group. All the work I've told you with the telomerase gene therapy has been done in collaboration with Fatima Bosch, that she's an expert in telomerase gene therapy. And this is the funding of the lab. I hope I was not too long, and I hope everything was clear. Sorry if this was too simple, because I know that you know my work, but I wanted to give you an overview of what we are doing. Thank you.
Thank you very much, Maria. And it was not too long, and I think it was perfectly appropriate because not everyone is a specialist of the questions you were mentioning.
Thank you.
So yeah. So for the Q&A session now, that will start now, I will ask a participant to raise their hands. So you can do that in the chat room, I think, or send a message if you want, and I will list. But there's an easy way. If you have a small, is it on? Yeah, you have a small arrow, I think, somewhere, and you can raise your hand if you know how to do that.
It's in participants.
Yes.
So bottom of the screen, participants.
Thank you, Thomas. Yeah. Okay. So Gregor, you have a first question, Gregor.
Thank you very much. This was a very exciting overview talk of your work. I really liked it a lot.
Thank you.
Let me ask a rather naive question, but the answer might in fact be very tricky to get hold of. So if it seems that there is a lot of advantages of having telomerase active and the longer telomeres, why then is it turned off, the enzyme in the adult organisms? Why don't we see more organisms that maintain that level of telomerase activity as adults? Is there something adaptive to shutting it off, or is it just the selective shadow? What are your thoughts on that, given that maybe the standard answer many people would expect would be it's a trade-off between cancer and longevity, but your results also show that in terms of cancer, surprisingly, it's apparently not playing with fire too much to enable other cells to become cancerous, given that they have the longer telomeres.
Yeah, this is a very important question, and I thought about it many times, so I'm not sure I know the answer. But there are organisms like the snake mole rat, for instance, that has very high levels of telomerase and also of the telomere protective genes when they have sequenced the genome. So they are living longer, and they basically are telomerase transgenics, the snake mole rat. So they really express telomerase at very high levels. We have certainly shown, I think very clearly, that having longer telomeres than normal is not going to increase cancer. We didn't show that with the telomere gene therapy, because there we are putting telomerase late in life, and telomerase is being diluted. But with the long telomeres, with the epigenetic experiment, the one where we generate chimeras from ESLs with hyper long telomeres, and we generate mice that are born with much longer telomeres than normal, and we have shown that these mice don't have more cancer. I think is a very clear demonstration that having longer telomeres is not bad. It's not going to increase cancer. Why we don't have longer telomeres? So maybe it's because we don't need, we didn't used to need them that long. I think there is an adjustment probably of telomere length and the time we used to last in nature, or species used to last in nature. So we in nature used to last, I don't know how, 30 years or 20 years. Or, we have plenty of telomeres for that. So we have plenty of telomeres to be perfectly healthy during 40 years, right? Is when we live beyond 40 years, when probably we would need more telomeres in order to prevent aging of the organism, which, and the same may happen in other organisms, right? So it's interesting to think about this paper I mentioned of the zoo, right, of different species, where you can see that the rates of telomere shortening, you know, vary a lot, and you can have species that are very distant in evolution genetically, let's say, but they share this rate of telomere shortening, and they share the same longevity. So somehow this must be saying that there must be a fast way to correlate let's say, what you last in nature with longevity, not even with your genes, which may take much longer because this is, but with changing something like the rate of telomere shortening. So I wonder whether also having telomeres that are adjusted to the survival in nature could be also a way to get rid of the less fitted individuals, if you want. I don't know. This experiment has not been done, but could be also a way to eliminate in a fast way individuals under stress conditions. For instance, we know that different stresses can be shorten telomeres, right? Maybe if we will have much longer telomeres, these stresses will have a lower impact on our survival. I don't know. This is all hypothetical. I have no idea, basically. Why is that?
Thank you. So we have another question by Jean-François. Jean-François, you have to reactivate your microphone so that we can hear you.
Okay, thank you. You can hear me?
Yes, we can.
Good. So thank you very much, Maya, for this nice talk. And I have a lot of questions, but I will focus only on one aspect. Is that you told us and you showed that there is an obvious link between the telomerase and the stem cells.
Yes.
So could it be that we could hypothesize maybe that in fact what you observed in your experience and your mice and so on is that you alleviate somehow the use up of the stem cells. So instead of consuming stem cells in normal mice because they are using up their telomeres, you are protecting those cells of doing so. So you are sparing your capital of in stem cells. So did you try to measure for example, the number of senescent cells that are in those mice, and how does it could intersect with that aspect of aging?
Yeah, it's interesting what you're saying. So this would mean that that by you know, that the stem cells somehow are not part of this effect when we elongate the telomeres. But we don't think that's the case because we have measured telomere shortening with aging, and this also happens in the stem cells. So when we generate these mice with the hyper long telomeres, for instance, we have seen that telomeres shorten at the same rate than the normal mice. So
exactly.
The shortening mechanisms are the same. Is that they start off with longer telomeres because they start off with longer telomeres also in the stem cells. They last longer, but the stem cells also shorten telomeres. What we think happens is that by having longer telomeres, the stem cells, you maintain for longer times the pool.
Exactly.
The pool
and
of
the potential of being a stem cell.
I agree with that. Yeah.
So, okay, good.
And regarding senescence, I think in the paper with the mice with the hyperlong telomeres, the 100% chimeras, we have looked at I think P16, P19. I'm not sure if I recall properly, but in general, yes. When you have longer telomeres, you have less activation of P21, P53, which are important pathways for triggering the senescence. And
so therefore,
in the lung model, where we have pulmonary fibrosis owing to short telomeres, there is an increase in senescence in the alveolar type 2 cells, and we can revert that by activating telomerase in the lung, in the alveolar type 2 cells.
I'm very interested in this type of experiments, the fibrosis, because we can then, from this starting point, we can hypothesize also that aging is not merely the result of shortening telomeres or blocking telomerase to restore, those telomeres, but something else that will end up with the using up of the stem cells, and therefore having this need to restore the telomeres to prevent this the use up of the stem cells. So in your model of fibrosis, you quote that in your one of the slide, a collagen deposition, but could you precise a little bit more about that particular point? I mean, did you observe less fibrosis, and which type of fibrosis, and how does it organize, and the connection with the immune system, of course, because it's connected with the repair mechanisms. And
yes, we describe all that in the paper, in the eLife paper, 2018.
Okay, I will read it.
We have looked at collagen by looking at collagen peptides, by looking at fibers, by PCR, different collagen types. So yeah, there is less production of collagen, and there is also, we have done also a panel of inflammatory cytokines, and there is also a reduction of many different pro-inflammatory factors in inflammatory cytokines. So all this is, I didn't put it here because I wanted to talk about maybe too many things. But this is described in the paper. So there is less inflammation, less activation of pro-fibrotic pathways, and because we have also done gene expression studies we have isolated the alveolar type 2 cells and studied the expression of genes in the ones that express telomerase or they don't express telomerase, and it's very clear that we see less activation of the pro-fibrotic pathway, the TGF-beta pathway, and then also we can detect less collagen by different techniques.
And less death also.
Jean-François, we will keep your question for the private conversation good. We have later. So if other participants want to ask a question, you will be able to ask yours just after. We have time for, let's say we have time for one last question. Okay, is anyone raising their hands or no? Okay, so maybe I will ask a last question. Very simple, a short one. Last month there was this paper published about results on senolytics and secondary effects of the use of senolytics as a way to delay aging. I don't know whether you have had the time to see it, Maria, but and one of the effects, the bad effects of the treatment that was on the test, and I can't remember which one it was that we had precisely liver fibrosis, severe liver fibrosis. So do you have any explanation or hypothesis about why it should have occurred?
Yeah, well, I'm a little bit skeptical with the senolytic approach to treat age-related diseases. And the reason is the following. Senescence, I think senescence and apoptosis, as you can see in this slide, or DNA damage, I think they are downstream of whatever damage is causing or impairing the ability of stem cells to regenerate. So if you would remove senescent cells, you still have upstream events that are causing the damage. Could be short telomeres or whatever that is impairing the stem cells to regenerate. So even though maybe senolytics may have could have some effect, I don't think they are going to be able to cure a patient with a degenerative disease. It's like the same as with the inhibitors of the TGF-beta pathway or that there are now. They are acting downstream also. So they are acting to prevent fibrosis, and they don't cure the patients because you still have the origin of the disease there. You still have the cause. Or the same with the anti-inflammatory clinical trials that have been done, for instance, for pulmonary fibrosis, they also failed. So I think anything you do downstream of what is at the origin is not going to cure. So I know the Mayo clinical trial with senolytics. I don't know if this was this paper because I don't know which paper I'm referring to, but at least this clinical trial is failing. I mean, I heard a talk about the results, the preliminary results, and it's not working. It's not curing the patient. So I think it's an interesting hypothesis, the senolytic hypothesis. But in my view, this is downstream of the real origin of the disease. So if you want to act, you have to act upstream. It's like trying to cure AIDS by trying to remove the sarcoma Kaposi. I mean, the Kaposi sarcoma, you need to go and kill the virus, right? So it's the...
Okay, I see. I get the answer, and I think I quite agree with you. So thank you again, Maria. Thank you for everyone for their participation to the conference. And so I will end the conference now, except for the small group, for our small group. And thank you again for the talk and the public talk.
Bye to all those of you that are leaving. Thank you. It was a pleasure.
Thank you very much.
Bye.
You.
Okay, we'll just wait a little bit for people to have the time to disconnect.
Je voudrais augmenter, moi, là.
Okay, so I have to stop sharing my screen also.
Yes, you can. Yeah. And okay, I think, okay. Okay. And I think the people that are remaining are all people, yes, all people from the group. So first of all, maybe Maria, if you want to take a very short break before we have the conversation, of course it's
no, it's fine. I have water here.
Okay, perfect. Yeah, I will. So first of all, we will introduce ourselves. So these are the people that are remaining are people from the group. You don't have everyone from the group, but you have